somatic. And somatic, remember, we're talking about the parts of the body that you're consciously aware of. So skin and muscle and bone experience fast pain. We're talking skin, muscle, and bone. You break your bone, you cut the skin, you tear a muscle, that's fast pain. And, or that can do fast pain. Not always, it can be slow pain, but you don't feel fast pain in your viscera. So if you were having surgery without anesthesia, you would feel the fast pain of the incision in your skin and muscle, but once they got into your internal organs, you would not feel fast pain. You would feel only slow pain. And slow pain travels at a slower rate and is different in some other ways. So fast pain is always going to be in those conscious parts of your body that you can feel, that you're consciously aware of, and that would be skin, muscle, and bone. Opioids like morphine are not effective against fast pain. What oftentimes works, oddly enough, is rubbing or putting pressure on the injured site. What that does is it stimulates other receptors in your skin or in the structure, and they start sending signals to the brain, and it kind of distracts the brain from that pain stimulus. So it's not really fixing the pain or anything, it's just kind of distracting your brain from it. But rubbing and pressure, you might notice, do help when you feel fast pain in your skin or muscle. Now, slow pain, on the other hand, is transmitted through small diameter unmyelinated nerve fibers. So those nociceptors have smaller diameter. Smaller diameter transmits more slowly than or large diameter. So fast pain, bigger diameter than slow pain fibers, and also they're unmyelinated. So the combination of having small diameter and no myelin sheath means that it's going to transmit a little more slowly. It's still, relatively speaking, you're still transmitting impulses quickly, just not as fast. And so it is more like, instead of being a 0.1 second, it might be a second or more after the injury. And at first it may not hurt as much, and then it starts to hurt more. So it's like it intensifies and intensifies over time. So if you stub your toe initially, hitting your toe really hard on something hard, maybe even breaking your toenail or cutting yourself, would stimulate those fast pain receptors, and that pain would shoot to your central nervous system, and you might even have a reflex. You might pull your foot up to, in response to that. And then your brain's going to process that as fast pain. But then your toe continues to hurt and throb. It might hurt for a while, and it seems to radiate or seems to throb. That would be slow pain. Slow pain can be chronic, and by that means fast pain is acute or quick. Chronic pain, which is more long-term pain, is slow pain. And so if it's hurting for a while, that's slow pain. Also, just if you have a burning sensation in a part of the body or an achiness, that is also, usually slow pain. Slow pain often radiates, so it might start out in like a muscle and then kind of radiate out and feel like it's spreading. So it doesn't always do that, but sometimes it does. And sometimes, like I said, it throbs. Now, slow pain can happen in your skin, it can happen in muscle and bone, but it can also, and if you, for instance, you're exercising and your muscle gets sore, unless if you tear a muscle or something, that's going to generate fast pain. But just exercising, that pain of that muscle pain is slow pain, and that can last for a while. But it can also happen, slow pain can happen in internal organs. So the pain you feel inside, in your gut, or in your heart, in your lungs, that kind of pain would be slow pain. You can't feel the cut of a knife in your gut, but you can feel other types of pain in the gut. So oddly enough, if you had surgery, you'd feel the pain of the incision in your skin and muscle, but you wouldn't feel the incision in your intestines, let's say. But you might end up with some slow pain after the surgery. You might, as you're recovering, you might feel internal throbbing pain, which would be slow pain. Now, slow pain can be treated with opioids like morphine. So if you had a backache, if you had a back injury, you had, let's say, a compressed disc compressing the nerve, that would be probably slow pain that you're going to be feeling, and that can be treated with opioids like morphine. So morphine can work on slow pain, which would be like the internal pain after surgery and things like that, but it can't really work for fast pain. It doesn't seem to work at all. Now, fast pain is localized. You can feel where it's happening. If you prick your finger or you get a needle stick in your arm, you feel where it's happening. You know it's there. And sometimes it does. Sometimes, well, that fast pain is localized. Let me just say that. But somatic slow pain, which would be in your skin and muscle and bone, that can be diffused, or it can be radiating out, but you have a general idea of where it feels. You can feel it, where it is. So if your muscle's sore, you can rub that muscle. You feel where the muscle is hurting, even though it may be more diffused pain. So somatic slow pain can be localized. Visceral pain, which is the internal organs, always slow pain. And you get visceral pain when you have things like injuries and trauma and surgery and things like that. Obstruction, like if you have gallstones or kidney stones, that can create slow pain. Also a heart attack, which is obstruction, oftentimes obstruction of a blood vessel, an artery, leading to a heart attack. That would be, a heart attack would be slow pain. Labor pains during delivery of a baby, that's slow pain. Now, visceral pain can be felt in the area where it's happening, but it can also sometimes be felt in other areas, and that's when it's called referred pain. Oftentimes, the slow visceral pain that you feel feels like it's happening in one place, but it's actually originating somewhere else, and that's called referred pain. And that's what we're going to talk about very briefly. Sometimes the referred pain may be in the skin and structures just superficial to the structure, just like just to the outside in the body wall of the structure, or it can be fairly far away from the organ, and that's what we call referred pain. Your brain is interpreting the pain as being in one location when it's actually possibly happening in a different location, which can be confusing if you're trying to figure out what's wrong and what's causing the pain, because it may feel like it's happening in one place, but it's really happening, the pain is actually originating somewhere else. And for example, some people, when they have a heart attack, you might feel pain in your chest, and it might radiate down your left arm, not your right arm, but your left arm. But some people experience a heart attack by feeling it in their jaw and in their teeth. And that's where they first feel the pain, not in their heart, where you would expect, but in their jaw. And but most of the time it's like radiating down the arm. But referred pain can be weird, especially with heart attacks. And with women, sometimes that pain is different than it is in men, which is why women often ignore pain from a heart attack and then often die as a result, because it doesn't follow the same, scenario that men experience, which is typically radiating down the left arm. So here are some places where you might feel referred pain from a injury or some kind of problem in the body. And this picture's a little bit, I need to shrink it up a little bit because you can't read all of it. But for example, your appendix, the pain may end up being in this you may feel the pain in this area, not just in what the appendix is, which is approximately here, but it could feel like it's in this area or in the skin or body wall of that area. So that would be an example of referred pain. In the heart, it's usually felt in the chest and radiating down the left arm, but you also may feel the pain in your back. And what they don't show here is sometimes the pain is felt in the jaw. So you can see different places, like for instance, gallbladder, pain is kind of can be diffused. It feels like it's just, the right side of your upper right side of your abdomen, for example. The stomach, your stomach is right here, so it's just a little off, but still, stomach pain may be felt in the middle of your abdomen instead of to the left of the abdomen midline. And so you have different, this is some examples of referred pain. Kidney pain can be quite diffused. all through the lower back area. It might feel like you've got back pain when in fact it's kidney stones or something like that. Now the reason for this is not well understood. There's a lot of theories, or I should say hypotheses and explanations, but it seems to have it's partly due to the fact that these sensory neurons, these proprioceptor neurons, travel to the spinal cord through the same nerves that serve these dermatomes. By dermatomes, we're talking about skin segments that most of the sensory fibers travel through certain nerves in those different skin segments. So at the very front there, the front of the face, the trigeminal nerve, which is cranial nerve 5, sensory information coming from your face travels through the trigeminal nerve. So the face is when I'm touching my face, that's traveling through the trigeminal nerve, and that would also be pain sensations would travel through the trigeminal nerve. But the back of my head, like right here, is most of that sensation is traveling through the C2 nerve, and then you can, a little further down C3, and then a little further down C4. So each segment is a dermatome, and it is primarily innervated by those nerves. It means it's going into the spinal cord at that point. Now there is some overlap because of plexuses, if you recall those nerve plexuses, nerve networks. So there is some overlap. You might have some of the sensory fibers going through T11. Some fibers may actually, travel through T12 or T10, but this is primarily where the sensory fibers are traveling through that nerve. So it turns out that those internal organs, like the heart or the gallbladder or the appendix, those sensory fibers travel through the same sensory fibers for that particular dermatome where you're feeling referred pain. In other words, if you go back to this picture, the heart and the arm here, let's see where that is on here. So let's see, this dermatome, notice that T1, 2, 3, and 4, also, the dermatomes extend down into the arm. Oops, I'm doing the wrong side, but oh well, because he's facing back. But anyway, those nerves that travel down the arm, those are T1, 2, 3, 4, 5, and that's also where the nerves coming from the heart, the sensory nerves coming from the heart are traveling. And when the brain interprets that slow pain information, it almost seems to be misinterpreting based on where the nerves are coming from. So it's interpreting that pain is coming from the body wall or the skin and not from the organ itself. And they don't really know why this is, but there's some kind of perception of pain that's due to the fact that you've got these somatic neurons coming from those parts of the body, and those autonomic visceral receptors coming from those, coming through those same nerves, and it's like the wires are crossed somehow, in a sense, so that the perception of it is that instead of having pain in the heart itself, you're feeling the pain in the body wall around the heart, or instead of feeling pain specifically in the appendix, you're feeling it in a kind of a general area here, because the brain is interpreting it that way. So it has something to do with how that slow pain is being interpreted by the brain and where it's coming from and what dermatome or what nerves it shares with those, those dermatomes. Because the pain often feels like it's happening in the body wall of those dermatomes. Hopefully that makes sense. But again, it's not really well understood why this happens, but it is a fact. So when you're having to diagnose an illness, or what the pain is coming from, you have to keep in mind the referred pain map here, and think about that when you're trying to figure out what's wrong with somebody. Any questions about that? Okay, so proprioception, detecting changes in body position, not just that you're standing or laying down or upside down or whatever position you're in, but also changes in body position, movement. And this is necessary to coordinate balance and movement, because if if you don't know what position you're in, how are you going to maintain your body posture? How are you going to maintain or coordinate your movements and not be flailing around, uncoordinated? So proprioceptors are important for helping us coordinate our movements and helping us keep our balance so we're not leaning off to one side and falling down and things like that. Proprioceptors also help you discriminate weight so that you can, without looking, you can feel how heavy something is in your hand. You can tell the difference between an orange versus a heavy book or something like that. Even if you don't feel, if you're not even looking at your hand, you can tell the difference in weight. So you can assess the weight and therefore determine how much force you need to move that object. So you're kind of weighing it with your proprioceptors, and that helps you know how much muscle power I need to lift something heavy versus something light. So lifting this is going to require less muscle power than lifting a heavy book or heavy weight. The way I know how much, I can feel how much this weighs. Just put it in your hand, you have that sense of weight in your hand. That's proprioception. So, just hold your hand out here, wiggle your fingers. You know you're doing it, right? You can feel it out there. It's proprioceptors that allow you to do that. So that you're consciously aware of what's going on throughout your body, even if you're in the dark or you're blind or your eyes are closed. There are three major categories of proprioceptors: muscle spindles, tendon organs, and joint kinesthetic receptors. Muscle spindles, and most of these proprioceptors are slow adapting, though some are fast adapting. But most of these are slow adapting, meaning they continue to be stimulated, they continue to generate action potentials, and they might slow down, but it takes a while to adapt to that sensation. Muscle spindles are found in your skeletal muscle, and they measure stretching of the muscle. And then this information is monitored by your cerebral cortex so that you can monitor the stretching of a muscle and respond to that stretching. So you might change body position if there's too much stretching. And if you're stretching like you're stretching after exercise, you can feel that stretching that's happening in the muscle, and you don't want to overstretch because then you can tear a muscle. So this helps you in controlling and coordinating movements. This is also important because the cerebellum, and we'll talk more about the cerebellum in a minute, but the cerebellum is important in coordinating muscle contraction. So the cerebellum monitors proprioception. That's one of the things it monitors. It's going to monitor the stretching of muscle. It's going to monitor the movement of muscle. And so this way it can coordinate the movement so you have coordinated and not kind of random movements happening, that your muscle movements are very coordinated and controlled by the cerebellum. This picture shows a muscle and then a tendon attached to that muscle. And there you can see the picture from where this was taken, the arm. And this is a muscle spindle. They're found throughout the muscle, not just one. It kind of looks giant size. They're really not going to be this big. They're going to be microscopic structures. But notice that, and we're not going to worry about the motor fibers in this picture, but the sensory fibers shown in yellow and blue, those are the sensory fibers transmitting sensory information, stretching information to the central nervous system. You can look at the key there. Red means it's going to the central nervous system. And notice that the nerve fibers are wrapped around, these are dendrites that are wrapped around muscles within the muscle spindle apparatus. So you have some of the muscles, not all of your muscles, but some of the muscles in your muscle are wrapped in these receptors, these free nerve endings. And when the muscle is stretched, when it's being pulled that way, not contracting but stretching, these get stimulated and they're going to send action potentials to the central nervous system. There are also some motor fibers associated. I think the one in red is a motor fiber that sends signals to the muscle. We're not going to worry about the motor fibers, but there are motor fibers that go to these spindle apparatuses as well. You can read about them if you want to in the textbook, but it gets kind of more complicated than I really want to get into for this. But that's what these are detecting, is stretching of the muscle. And then you have what are called tendon organs. These are encapsulated. These are they almost look like synaptic end bulbs, but these are supposed to be dendrites, and they are in a capsule, like the Pacinian corpuscles in your skin and the Meissner's corpuscles. These are encapsulated in a little capsule of connective tissue. They are located in the tendon, close to where the muscle and tendon come together, and they respond to muscle contraction. So while these are responding to stretching, these are responding to contraction of muscle. In other words, if I'm stretching a muscle, then that's going to be stimulating those spindle fibers. When I contract, you might notice that when you contract a muscle, you tighten the tendon. You can feel the tendon tighten. That's being picked up and stimulated by, or that's going to stimulate the tendon organs. So tendon organs are described here. They are encapsulated dendrites, and they just respond to tightening of the muscle. Again, this is sending signals to your brain, especially like the cerebellum, so that you don't over contract, you don't over tighten the muscle and possibly tear the muscle as a response to too much tension. So this way you can kind of control the amount of tension necessary for a particular movement. If you're walking, you only need a certain amount of muscle contraction to walk versus running. Or if you're lifting something lightweight, you'd only need a little bit of contraction versus something heavy. And you don't want to over tighten the muscle unnecessarily if you don't need to. And that could, in some cases, lead to injury of the muscle. So this just helps protect the tendons and muscles from injury. And then you have the joint kinesthetic receptors, and these are actually a bunch of different receptors. Free nerve endings, Ruffini corpuscles, and Pacinian corpuscles found in the articular capsules of synovial joints. So I inserted a picture here, remind you of what a joint, these are synovial joints, which are the joints that have synovial cavity with synovial fluid in it, and they're enclosed in a capsule of dense irregular connective tissue called the articular capsule. And so this capsule encloses this synovial cavity, and where the synovial fluid is. And it helps hold the bones together, it helps enclose that joint space. These joint kinesthetic receptors are found in this connective tissue of the articular capsule. The free nerve endings and Ruffini corpuscles respond to pressure at the joint. If there's any kind of pressure being put on the joint, they respond to that. Ruffini corpuscles are also found in the skin, so of course with free nerve endings. And Ruffini corpuscles are involved in stretching, like in the palm of your hand and soles of your feet. They're stretch receptors, but in the joints, they're responding to primarily to pressure at the joint, like when you're standing on your knee, standing on your legs, you're putting pressure on the capsule of your knee joint. So they would be responding to that pressure. Pacinian corpuscles, which are also pressure receptors in the skin, respond to pressure in a different way. They're responding to the pressure that happens during acceleration and deceleration. In other words, acceleration means when I start moving a joint, and deceleration is stopping or slowing down. They're responding to that. So, if you move your arm, you can feel it, moving. Part of that is because of those Pacinian corpuscles that are detecting that change in pressure due to movement at the joint. So they are detecting that and sending that to the cerebellum. All of these, notice that these proprioceptors are all located either in muscle, tendons, or the joint capsules. So they are detecting, what's going on in the muscles, in the joints, in the tendons during either when you're staying still or when you're moving. And that information is going to the brain, specifically to primarily the cerebellum, and the cerebellum is taking that information and just determining what's the appropriate response. Should I contract more or less? Should I stop you from stretching? If you're stretching too much, things like that. We're going to come back to the cerebellum shortly, but first let's look at something called receptive fields, which has to do with how sensitive your skin and other structures are. It's based on the number of sensory neurons innervating the skin or other structures. The receptive field is defined as the area of the body in which stimulation leads to a response in a specific sensory neuron. In other words, let's go to this picture. I've just put this picture in, so I'm going to repost the notes. Every sensory neuron, so these are showing free nerve endings. It's not always free nerve endings, but it's easier to show with free nerve endings, I guess. But each sensory neuron, here's one over here, here's one over here. This is supposed to be the skin, and these are supposed to be free nerve endings, which are dendrites in the skin. This is the epidermis and dermis and subcutaneous layer. This is the sensory axon here, myelinated, okay? But here's the dendrites, where they are being stimulated by either touch, free nerve endings, not so much touch, but pain, temperature, or something else. But you do have touch receptors, and they also have their receptive fields as well. Notice that this sensory neuron, its receptive field is in this colored area here. That's where the dendrites are penetrating, so that's where it's going to be stimulated. And then here's another sensory neuron over here, and its receptive field is over here. And this sensory neuron's receptive field is over here, okay? So they're not really overlapping. The receptive fields don't overlap. If I touch the skin here or here, I'm going to stimulate this neuron. It's going to feel like the same point. No matter, I could stimulate it here in this side of the receptive field or this side. It's still going to feel like the same point is being stimulated. It won't feel like two different points. And I'm going to show you that in the threshold test in just a minute. But if I touch here and then I simultaneously touch here, it'll feel like two different points there. So you can feel those two separate sensations in the skin. But if I touch in the same receptive field, it'll feel like the same sensation. It'll feel like it's one touch instead of two. Yeah?
Do free nerve ending cells have a broader range than encapsulated cells?
It would seem so, yeah, because those Pacinian corpuscles are encapsulated, and so are Meissner's corpuscles, but I think they're stimulated, they're mechanically stimulated, and so that means that, let's say you've got this Pacinian corpuscle or Meissner's corpuscle, and you're stimulating over here, that's still deforming this, it's still putting pressure on it or touching it, it can still be sensed. So it would be there and the area, around it. But I don't think, it's hard to, I'm not sure exactly how big that field is. So, it would be that field immediately around it, and another really big field around it. So you'd have to have other Meissner's corpuscles taking care of those other receptive fields. And speaking of that, if you have fewer sensory neurons in a part of the body, each sensory neuron is going to be in charge of a larger field. If you have more sensory neurons, each sensory neuron is going to be receiving information from a smaller field because they don't overlap, their fields don't overlap. So the field size is inversely related to the number of sensory neurons. Let me explain what that means. Okay, so each sensory neuron innervates a specific region, a specific sized area. And the receptive field size varies inversely with the density of sensory receptors. In other words, the more sensory neurons there are, the smaller the receptive field. And the fewer sensory neurons, the bigger the receptive field. So you're going to have, if it's more sensitive skin, you're going to have more sensory neurons with smaller receptive fields, right? And if it's less sensitive area of the body, you're going to have fewer sensory neurons, and those neurons are going to have larger receptive fields. So it's an inverse relationship between the size of the field and how many sensory neurons are in that area of the body. So in other words, if this is a less sensitive area, you're going to have fewer of these, but they're all going to be larger fields. If it's more sensitive, you would have, these fields would be shrunken, and you would have more sensory neurons filling in those spaces there. You would have more of those. So for example, if you look at the skin on the back of your leg, on your back and on your legs, those areas of skin have fewer tactile receptors, meaning touch receptors or pain receptors or thermoreceptors or any kind of, tactile meaning touch, not thermoreceptors, I'm sorry, or pain receptors, but touch receptors, but also pain receptors and thermoreceptors would apply to this. I really should take tactile out because it could be any variety of sensory receptors, but anyway. In your skin, on your, on your back and your legs, you have a lower density of at least the tactile receptors. And that means that their receptive fields are larger and those bits of skin are less sensitive. Now that is tactile. I will say this, your back does have a high concentration of cold receptors, so when cold water hits your back, you really feel it. So does your stomach area. So even though your back and stomach are not that sensitive to touch, they are sensitive to cold. And you do have a fairly high density of cold receptors in those areas. You might notice that you feel cold more in your trunk than you do maybe in your legs because there's more cold receptors. So they would have smaller receptive fields and more of them. But when it comes to touch or tactile sensations, the skin of your legs and the skin on your back are less sensitive. They have fewer sensory receptors and they're larger receptive fields. On the other hand, the sensory receptors or the skin of your face and the skin of your fingers and hands have a very high density of sensory receptors, and their receptive fields are much smaller because those are sensitive regions of the body. So the way you figure this out, and you can do this at home, you can do your own series of experiments if you want to, determining the 2-point discrimination threshold. This gives you an idea of the relative sizes of receptive fields in a part of the body, in the skin, in different parts of the body. And what this is, the 2-point discrimination threshold means the minimum distance that 2 points can be discerned as 2 separate points and not one. Okay, and I'll show you a picture to explain that. And we'll come back to this in a second. So let's say you're using these forceps to stimulate the skin. They've got points on them, but you could do this with 2 pins or 2 pencils or something like that. You don't need to prick the finger or anything like that. You just need to stimulate the skin enough with touch. And it has to be something pointy so that you can discern that as a point and not a kind of wide area or anything like that. So if this is one sensory neuron, and here's the nerve endings of that neuron, and you poke the skin in 2 places, but it's within the same receptive field, it'll feel like 1 point. If you're not looking at that, and somebody does that, it would feel like 1 point, not 2 points. Because whether you stimulate here, 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 or here, you're stimulating the same sensory neuron. Your brain's going to interpret that as 1 point. But if you poke the skin in 2 receptive fields of 2 neighboring sensory neurons, so this neuron serves this field, this neuron is innervating this field. If you prick the skin here and here, within that 1 field, it would feel like 1 point. But if you prick the skin here and here, it'll feel like 2 points, okay? And so if you do that, like do that on your arm or your leg, your back, your hand, the back of your hand or the front of your hand or your fingertips, you'll find that, if it's your back and you have somebody else do it so you don't know, because if you know, then you kind of start second guessing yourself. But you have one other person do this to you, put like forceps or something that you can space out. It has to be something like pins or something you can space out in different amounts. But if you're not looking at that and you have somebody else do that, you tell them, okay, I feel 1 point or I feel 2 points, and you can kind of, measure that 2-point threshold. So like it says here, the 2-point threshold, discrimination threshold is about 3 millimeters, very tiny, in your fingertips. In other words, the point where you feel not 1 but 2 sensations in your fingertips is only about 3 millimeters. And by the way, these forceps, these are basically calipers. You can measure the distance with these. Of course, you have to kind of do a more rough estimate if you're doing it at home, unless you have calipers. But calipers work great because they tell you the exact measurement. In your forearm, it's a much wider 2-point threshold. In other words, the point where you feel 2 points without looking is a much wider distance, it's 40 millimeters. Now, 50 millimeters is about 2 inches, so that's quite a distance there. So anyway, that's how you figure out the relative size or the approximate size of the receptive fields by doing these 2-point discriminations. You start out narrow, and if you still feel 1 point, you make it a little wider and a little wider each time, and you try to see how, what point do you feel 2 points, not 1. That's the 2-point discrimination. Any questions about that? Like I said, you can do that, as long as you have a partner, you can do that with, on your own body. Check it out for yourself. Next thing we're going to look at is somatic sensory pathways. And these are the neurons in, and by somatic, again, we're talking skin and muscle and bone, structures that you can feel sensations in at a conscious level. It goes through the cerebral cortex. And there's 3 sets of neurons sending signals to your brain. First order neurons are sensory neurons, and I'll have a picture of this in just a second, but first order neurons are sensory neurons that transmit impulses from these various somatic sensory receptors, touch, pressure, pain, and proprioception and all that, thermal receptors. Any of those that we've been talking about so far are somatic receptors. And this information is sent through these first order neurons, which are sensory neurons, and this information goes to the brainstem or spinal cord. And these neurons then synapse with the second order neurons, which are interneurons. And these guys usually send impulses to the thalamus. Most sensory information goes first to the thalamus through the second order interneurons. And then the second order interneurons synapse with third order interneurons that send information to specific parts of to specific sensory areas. So far, what we've been talking about, somatic sensation, touch, pressure, pain, temperature, heat and cold, proprioception, they all are somatic and they all go to the same area, the primary somatosensory area, which we're going to be looking at as well. So, first of all, this picture I showed you previously of these different unipolar sensory neurons that are either have free nerve endings or encapsulated ones like Meissner's corpuscles and Pacinian corpuscles and tendon organs, or the Merkel cells that stimulate this neuron. This is also a first order sensory neuron, not the receptor cell itself. The neuron that's stimulated by it is the first order sensory neuron. These are all first order sensory neurons in the somatic system. And so that's what this one is. This neuron, which is either a Meissner's or Pacinian corpuscle, is a first order neuron. First order sensory neuron, sending impulses to the spinal cord through this, in this case, through the spinal nerve, and then up through the spinal cord. Here it's synapsing. In this case, in the brainstem, it's synapsing with an interneuron. This is the second order neuron, the interneuron here that sends signals to the thalamus. The thalamus then sends signals to the primary somatosensory area. That one up here is a third order neuron. It's a third order interneuron. So this picture, shows you that somatic pathway. What the thalamus does, and I'm going to show you some other pictures of, this is a frontal section. The thalamus is basically a relay station for most of the sensory impulses, both somatic ones as well as the special senses. Many of those, all but one, pass through the thalamus first. The sense, we'll get into the one that doesn't later, but the thalamus functions as a relay station for most sensory impulses. And in the case of somatic senses that we talked about, it's going to send signals to the primary somatosensory area of the cerebral cortex. The thalamus is in the region of the brain called the diencephalon, which is this region here. It consists of the pineal gland over here. There's the thalamus and the hypothalamus here is attached to the pituitary. The red oval area, there's 2 oval masses deep inside your brain, that's the thalamus. And here's another frontal section. The thalamus is this red area right here. It's just below the corpus callosum and lateral ventricle spaces here where cerebrospinal fluid is. It's deep to the basal ganglia, which we're going to talk about those basal ganglia as well in just a little bit. But in this picture, and don't worry about the name of the pathway and all this precise information, I just want to show you the two ways that somatic senses can travel. This is showing first order sensory neurons, which are carrying information from the receptors that deal with touch, pressure, vibration, proprioception in the upper limbs, etc. Don't worry about, I'm not going to test you on where these go specifically, but we're just going to look at the two examples of how, or the three examples of how sensory information can travel. But you should know some general stuff that I'll be pointing out. So these receptors are sending signals through the first order neuron, which is a sensory neuron. Notice it enters through the posterior nerve root. There's the posterior root ganglion where the cell bodies are, these unipolar neurons. So those cell bodies, remember, from way back here, these are in the posterior root ganglions of the nerve root here, the dorsal nerve root. And the solid line represents the axon. It travels up through the tracts, the ascending sensory tracts in the white matter of the spinal cord, up all the way to the medulla. The medulla, by the way, is, remember the brainstem, there's the spinal cord, here's the medulla oblongata, above it is the pons, and above that is the midbrain. So that sensory information is traveling into the medulla, and it's in the medulla where this neuron, this first order neuron, synapses with the second order neuron, which is an interneuron. And this is the cell body, this is the synapse right here. There's the cell body of that interneuron. Remember, the interneurons are multipolar. The axon crosses over in the medulla, so you have crossing over of sensation from one side to the other, from right to left or left to right in the medulla, and then that information gets transmitted up to the thalamus, where there's a synapse with a third order neuron up there. Let me see if this light works. There we go. There's the synapse. It's kind of hard to see, but there's a third order neuron sending its axon to the primary somatosensory area, and we're going to be looking at where that area is shortly, but that's where that information is going. So you have 1, 2, 3, first, second, third neurons. First one's sensory, second and third are interneurons in the central nervous system. Here's a picture of a side view of the brain. This is anterior, this is posterior. This is the frontal lobe, parietal lobe, occipital, and temporal. In the parietal lobe, there is a ridge of brain, of cerebral cortex, called the post-central gyrus. It is posterior to the central sulcus, which separates the frontal lobe from the parietal lobe. So post-central meaning behind the central sulcus. That blue area has been, as we'll see, mapped out, and we know that somatic information of touch, pressure, pain, temperature, vibration, itching, all that stuff, including also proprioception, all goes to that primary somatosensory area and is perceived there. So here's another possibility. This is other types of receptors: heat, cold, touch, pressure, tickling, and itching receptors. They are sending their impulses in. This is the first order sensory neuron, but this synapse is not happening in the brain, it's happening in the spinal cord. So for some other receptors, sensation crosses over in the spinal cord. Here's the synapse with the second order neuron. The axon of the second order neuron, which is an interneuron, travels up to the spinal cord through the brainstem. It's already crossed over in the spinal cord, so it doesn't cross over again, and sends signals to the thalamus, and the third order interneuron is there sending signals to the primary somatosensory area. So depending on where the information is coming from, it either crosses over in the spinal cord, and that's where the synapse is between the first order and second order neurons are, or it crosses over in the brainstem. I don't expect you to know which is which or the name of this pathway, but do know that somatic sensory information crosses over in the brainstem or in the spinal cord, depending on where it's coming from. And also, very quickly, what time do I have here? Oh, I've got 2 minutes, hold your horses. Trigeminal cranial nerve. Remember I told you your face is receiving innervation from the trigeminal nerve, which is cranial nerve 5. That's what this is. There is a ganglion on either side of the pons. Now the pons, remember, go back to this picture back here somewhere. There's the pons. It looks like a little belly sticking out, the brainstem. And the trigeminal is attached to that pons. Information coming from your face enters through the first order sensory neurons. And the synapse here, there's 2 synapses. One is with a second order interneuron in the pons, or it could drop down to the medulla and cross over there. So it either crosses over in the pons or the medulla, and then that second order neuron, in either case, goes up again to the thalamus, and the thalamus sends that information through the third order neuron to the primary somatosensory area. So there is still crossing over. So when I'm touching this, it's crossing over in the brainstem to the left side. Or if I'm touching here, it's crossing over to the right side. So all somatic sensations cross over, either in the spinal cord or the brainstem, and either the medulla or the pons in this case. So we're going to finish this up on